Good morning, good afternoon, good evening to all. Today, there is a discussion. I invite Dr. Anil KHURANA sir, Dr. K.C. MURALEEDHARAN sir, Dr. ANIRBAN Sukul, and today we have a wonderful moderator, Dr. Shweta Singh. She will describe everything about you. Now I invite Dr. Shweta Singh to moderate this wonderful discussion. Okay, you are welcome. You can start now.
Hello everyone, I, Dr. Shweta Singh, welcome you all in the 1846th day of uninterrupted daily webinar of IFPH, which is an international homeopathic humanitarian organization formed to bring together the global promotion of homeopathy and to bring together homeopathic movements, activists, and activities worldwide under one umbrella. Before beginning the session, let's have a 1 minute silent prayer. Thank you, everyone. And today's panel discussion focuses on a theme of utmost importance, that is, challenges of research in homeopathy. The topic holds great importance for both students, practitioners, and researchers. For students, it provides clarity on how scientific evidence strengthens learning and practice. For practitioners, it highlights the need to adapt, innovate, and validate clinical experiences through research. Understanding these challenges bridges the gap between knowledge and applications of homeopathic medicines, ultimately advancing the future of homeopathy. And we are privileged to have with us our distinguished panelists today. Let me introduce them to all of you first. Dr. Anil Khurana, sir, who is the former Director General, CCRH, New Delhi. His way of functioning with sincerity and perseverance, with smiling simplicity of life, has stored in himself the vastness of experience and inspiration for all. His skill in good practices in clinical research bioethics has contributed for the development of research activities as CCRH and also in many of the homeopathic institutions. He has become the guiding light for the new generation to inculcate research aptitude in them. He is the former chairman of the National Commission for Homeopathy. During his tenure, he contributed to align research and education, recipient of numerous national and international awards. Besides his research activities at CCRH, Dr. Khurana sir is actively involved in health related services to the society and worked for several issues such as mother and child cares, HIV, and at present for the for COVID-19. Our next panelist is Dr. K.C. Muralidharan sir, who is BHMS from Mahatma Gandhi University, Kerala, MD from Maharashtra. Currently, he is the Assistant Director Homeopathy and Scientist for and Admin in charge of Central Council for Research in Homeopathy, New Delhi. Worked under different projects like behavioral disorders, mental retardation, and psychosomatic disorders, and HIV, AIDS, etc. He is the author of many national and international peer-reviewed journals. Our next panelist is Dr. ANIRBAN Sukul, who is BHMS, MSc Zoology, and PhD. He is director of Sukul Institute of Homeopathic Research, Kolkata, India, editor of Clinical and Experimental Homeopathy, author of numerous research articles published in international and national journals, and a book published by Kluwer Academic Publishers, the Netherlands. He has lectured on homeopathy in 14 countries across Asia, Europe, UK, and USA. He has been conferred the Junior Scientist Award India, Lifetime Achievement Award Bangladesh, Gold Medal Award UK, and many more. Recently, he was nominated for the Excellence in Integrative Medicine Research Award by the European Society of Integrative Medicine. So, these are our panelists. I welcome all of you. And now, let's begin the discussion with Dr. Khurana, sir. Considering the ongoing debate around the scientific basis of homeopathy, it is essential to explore novel ways to investigate its underlying mechanism. My first question to you, sir. Given that homeopathic remedies are ultra dilute, what innovative experimental models can be used to study their mechanism of action at a molecular or biophysical level?
Thank you, Shweta. And am I audible?
Yes, sir.
Okay, thank you. Thank you, Dr. Sani, for inviting me here for this IFPH talk, especially on this panel discussion. And this topic has been very core to my heart in the sense that I've been working on this fundamental basic set for more than 20 years in CCRH. And we understand what kind of difficulties we are facing at this point of time because of this mechanistic aspects of homeopathic medicine. Now, there are so much of challenges in taking up research, especially in the fundamental aspect. In the sense that homeopathic medicines are highly ultra dilute and they cannot be explained in terms of known laws of physics and chemistry. So they are way beyond the dilution factor, which is known in chemistry, that is Avogadro limit. And in that way, there's no existence of molecule which is expected, not to be expected in the high dilution, how these medicines are working actually. So in that way, mechanistic are a little difficult aspect when we talk about homeopathy. And that's why when we started exploring on fundamental basic research in CCRH at that point of time, in the year 2004 or 5, when the article in Lancet was published denouncing homeopathy as mere placebo. So that was basically a big jolt on the part of homeopathy, that such a big journal is publishing now an article against homeopathy. So we started working on these aspects. And as you have asked, what kind of approaches have to be taken up for explaining homeopathy, how does it act, what are the mechanism of action, and what are the pathways of action. And there's a long journey from 2005 onwards till the last year. More and more such studies have been taken up in collaboration with institutes of excellence. And the first thing we started working on was first to find out whether these medicines are not mere placebo or they are more than that. So in that way, we started working on some biological models. That is the first step which we wanted to first prove that these are not simple mere placebo. And then moving on to the mechanistic aspect. So very simple experiments of petri dish model laboratory experiments were taken up, whether homeopathic medicines are anti-infective, whether they can kill the bacteria, viruses, or they can inhibit the growth of those viruses. And if they are doing, they are not placebo, means we have to have active and positive and negative controls while taking up such studies. All those experiments have been published in different journals. And we were convinced after a lot of experiments, there is a decided biological effect of homeopathic medicine. And it cannot be said that these are simply placebo effect. Because we had a control of vehicle control also, we have a control of other comparative models like for antifungal effect, we had used ketoconazole and many other known antifungal agents, even antibiotics or even antiviral. Those kind of studies have been taken up. Now, after convincing ourselves that these have a decided biological effect, homeopathic medicines, we moved on to the next step of mechanistics actually, which we are asking now. What are different experimental models which we can take up for these mechanistic aspects. And we did looked into the literature review at that point of time. Many such articles have been published in the past. From Indian side, we know Dr. Khuda Baksh, who have been doing a lot of work to actually explore this aspects of homeopathic medicines. And he proved that homeopathic medicines, like for arsenic toxicity, they are in endemic population, and more people are consuming those arsenic to their water source. And there's a kind of a genotoxicity which is coming up to the population, and people are suffering from different kind of problems. And he did experiment on mice by creating a arsenic toxicity status, and then saw the effect of arsenic 30 and 200C, how these are actually removing those toxic condition into from the mice, and which epigenetic aspects which he worked upon, and he could prove that potentized arsenic reduced arsenic-induced genotoxicity in mice. And he even looked into the chromosomal aberrations, even mitotic index and sperm head anomaly also, which are usually induced by this arsenic toxicity. So these articles have been published in international forum. And thereafter, we also worked on cancer model with Bose Institute in Kolkata with Dr. Gauri Shankar Sir and Dr. Tanya Das, a scientist from this premier institute on basic research. And on cancer model, such studies have also been taken up to delineate the pathway of action of homeopathic medicine. And they also moved from biological response then to the mechanistic aspect, and they could also publish such studies in very high-end journals, PLOS One and other journals. So these studies are also came out with some kind of a suggestive way that yes, homeopathic medicines are actually in cancer disease in animal model studies as well as in cancer cell lines. That this proliferation of cancer cell lines would be halted by inducing a P53 stimulation through different enzymatic activity, which has been resulted into this apoptosis by P53 gene upregulation. That stimulation of P53 actually, and they have confirmed it through by knocking out that P53 from the cell line, that these our medicines were not working. So a mechanistic aspect has been worked upon, and for different cancer cell lines, different upregulation models have been worked out for lung carcinoma, breast carcinoma, and other tumors. So these have suggested different pathways of actions as well as different models which can work upon. Although there are so many of theoretical models which are known into the scientific world, when we started exploring the literature, you say we have been working on the hormesis model, and such studies have been published on hormesis, water memory by Ben Winist, and even electromagnetic signaling pathways by Luc Montagnier. He has suggested one of the hypothetical, although he has not worked directly on homeopathy, but he used samples on diluting DNA, bacterial DNA, and he proved that yes, electromagnetic pathways are there with homeopathic medicine. They might be emanating that electromagnetic signals, which actually through teleportation they can regenerate even bacterial DNA. Those kind of models have been worked upon by our premier scientists, and this is the summary which I could tell you on what kind of models or other different pathways which we can work upon, different kind of approaches which can be taken up. And let's move. We are thinking of with other scientists to work upon these areas where we can take up our studies. Thank you.
Very well explained, sir. Thank you so much. And my next question is to Dr. K.C. Muralidharan, sir. In homeopathy, as we need to maintain the standardized, individualized treatment in clinical research, so what alternative research designs can we adopt to evaluate individualized prescriptions without violating the scientific rigor?
Good evening, everyone. Respected other panelists and respected audience. Regarding this panel discussion on this individualized prescriptions without violating the scientific rigor. So that is the question asked by Dr. Shweta. And my assumption is that whether we need an alternative methods for this. So that is the first question we should ask, because why we need an alternative designs, because all the designs are already available in the domain, and why we need another domain in this research. We are assuming that we need a separate domain or alternative domain. It is because we are thinking that it's an individualized medicines, and we are evaluating the individualized prescriptions, and it is purely personalized medicine. So we think that actually in such conditions we don't get because it may not go with hand in hand with RCT, randomized controlled trial, or with the set guidelines of the international guidelines of the research. But when we are looking into the data, randomized controlled trials, the data available is on 2021. So we have completed almost 148 trials on 88 different medical conditions. That is in double-blind placebo-controlled trial, which we have completed. And the total number of randomized controlled trials, which we have conducted, is around 255 RCTs, which we have completed. So we are going ahead with all these randomized controlled trials. And again, whenever we are doing randomized controlled trials, again, we need to have some sort of issues with single prescriptions or individualized prescriptions of personalized medicine. It is, I personally believe that it is our mistake to choose the right design for the right study. Generally, we don't select the right design for the right studies. So that's why we don't get a right result or right outcome. Because we are looking at the mostly the scientists or the practitioners looking at the clinical research. So because most of the people are interested in clinical research. So as KHURANA sir said, actually, regarding the fundamental research. So that is one of the core area where we don't require any type of principled individualization for that type of research conditions. Because fundamental research is basically on the exploration of the basic principles of homeopathy. So which may not be required any type of personalized medicine, or which may not be required in individualized medicine. Rather, it required some sort of basic principle has to be proved, whether it is really correct or not. But when we are looking at the clinical research aspect, so we have already a lot of different models of studies are available. So I would say in a very brief, we have multi-criteria outcome studies that generally we do it. Generally we do it in multi-criteria outcome studies, which assess the multiple outcome relevant to the individualized treatment. So we are looking into such symptom relief, quality of life, patient satisfaction, all those things coming under the multi-criteria outcome MCO studies. So there is one model we can adopt it. Another model is NF1 trials. NF1 trials actually very rarely used in the medical system, but it is very close to homeopathy, and I have not seen any of the cases where we have tried this NF1 trials where we make a single patient and we treat those cases or intervention has been given to those cases in randomized manner, and we will be observing those cases, and their control will be the same control we are making it. So this is the short of NF1 trial. Another trial is you can adopt is the observational cohort studies with the propensity score matching. Again, you are creating some control of artificial control into this observational cohort studies, which is one of the ideal model for homeopathy area of clinical trials. And other studies like patient preference trials, then adaptive clinical trials. So adaptive clinical trials actually, it is a trial designed to allow modification to study protocol based on the accumulating data, because in between you can change the study protocol based on the what is the outcome we are getting it. Generally, this type of adaptive model is applied in the epidemic research. So same method we can adopt it. So this type of model we can adopt it. Another model is the multi-cohort longitudinal experimental MCLE designs. So this is generally we do it. Then some qualitative research we can do it, having focus group discussions and making some interviews and other things. Then you can have a participatory research approaches. You can make it where the engagement of the patient and practitioners can be made and make some outcome on that. So these are the few models already available in the research, and explore such research, because we are concentrating only on randomized control trials or some sort of a specific which are we are very familiar with that. So in order to make a different track of research interest, so we should try all those models. Accordingly, we can achieve our targets. So there is no specific model is required, but the models are available everywhere. Only we have to conceive those models and create our own protocol. Thank you.
Thank you so much, sir, for so clearly explaining research designs that preserve scientific rigor while maintaining the individualized essence of homeopathy. And my next question is to Dr. Anirban Sukul, sir. So, what large-scale epidemiological methods can be adopted to assess homeopathy's role in community health improvement?
Yes, good evening, everybody, and good evening, Dr. Shweta.
Good evening, sir.
First of all, I like to thank all the participants, all the speakers for taking part in this excellent session that has been chosen as a topic that the researchers challenges in the homeopathic research. This is a remarkably a challenge, of course, and it's a very deep-rooted challenge, whatever I can say, as far as basic research is concerned. Yes, of course, we have overcome, but I think before coming into my topic, I think you asked something. Can you please repeat? Did you ask me something, Dr.
Yes, sir,
Shweta?
sure. Sir, my question is, what large-scale epidemiological methods can be adapted to assess homeopathy's role in community health improvement?
Large-scale epidemiology, okay. See, epidemiology is occurred as not very frequent, of course, but did you ask me as the factors or what? Sorry, I can't hear you.
Sir, epidemiological methods that can be adapted to assess homeopathy role in community health improvement.
Because see, epidemiology is not such a disease because it's not very frequent that it comes everybody to every doctor. So in regard to that, with some maybe in some years or maybe in many years we can see such kind of epidemiology. Like COVID we have faced like this. So in such cases, of course, the method regarding to maintain the public community or public health, rather, in as far as homeopathic treatment is concerned, is something like that, there are some methods. If I take example of COVID-19, so there are some methods of that epidemiology, because COVID-19 isn't new to us or everybody. There are some methods has been adopted by the government of India. Now, these are, of course, as regard to the, not only for homeopathy, I should say, because in every scientific study regarding epidemiology, it should be assessed these are some kind of directly to the public community or the community, and these are also to the clinical, whatever we selected as a drug. So in regard to these, we should have a typical balance between the community, because not every community might not be facing the same epidemiology. So there are differences in having such kind of effects of epidemiology as far as the community is concerned. So in regard to the community, there are certain changes, of course, regarding the temperature, I mean climate or whatever. So there are certain changes as we can see, because tropical countries, they face such epidemiologies, like filariasis also is an endemic disease. And so this kind of things, those are based on the tropical region, may not be into the temperate zone, so in the cold countries. And regarding homeopathy, we have to go with the symptoms of all the collected symptoms of a disease. So it should not be biased to any kind of disease. Like, of course, we have found arsenic as a very good medicine for homeopathy as far as COVID-19 is concerned. But yes, of course, I personally have cured many COVID-19 patients not without giving arsenic album. So Bryonia and so there are many homeopathy medicines that are really remarkable to cure COVID-19 very successfully, as homeopathy treatment is offered. So I think this is the short answer. But what I discussed today, as far as the topic of this session of this evening, is that the challenges, of course, to what we face as a scientist. I personally have a background of studying MSc in zoology, and thereafter I got a PhD in neuropharmacology. I was the doctorate student, or the research associate under the University Grants Commission of the DSA, Department of Special Assistant, in Vishwabharati University in Shantiniketan. So I have a tremendous background of basic research on homeopathy. It's not directly, may not be sometimes during my PhD tenure, may not be sometimes directly related with the homeopathy. We had to work with the science of the homeopathy, because you can see I'm working under certain projects. So there have been some projects guidelines that I need to follow, or I had to follow rather. So, but of course we have obey with the science of the homeopathy, whatever it has. Now, during the tenure of my research, whatever I faced is that the drug itself, the dilution itself, the potency itself, because each and every potency, each and every dilution has the only just two things are there, is the ethanol and the water. There have been no other things, no other molecule, nothing, no other compound, nothing is there other than these two. So everywhere we found ethanol and alcohol, alcohol and water. So it's aqueous ethanol rather. So if it is so, now it is, of course. Now, if it is so, how can you differentiate between the potencies? How can you differentiate between the drugs? Because there is no drug molecule. So it's very, but as a disease, as a medicine itself, it affects as a differently. Different as a potencies and different as a drug. Of course, Naxfoam, sulfur, and so many so forth, they are differently acting, even though having same molecular, not molecular, but same solution or dilution, having ethanol and water, these two. So these two are the mainly differs. So that's the challenge I'm talking about, that I faced, or rather we faced, as in whatever Sukul Institute of Homeopathic Research does with the structural study of dilutions, that the hydrogen bond or the structural difference, whatever in short I can say, the structural difference is the main difference that implies to the water structure. So water structure differs as the potency differs, as the dilution differs. Now, in a water structure, we have 4 kind of hydrogen bonds. It's a hydrogen bond networks, hydrogen number of hydrogen bonds, hydrogen bond structure or the strength, and the hydrogen or the non-hydrogen bonded water or the free waters, whatever we say, what we say. So these are the 4 different, things we can get into a dilution or any solution of aqueous ethanol. So these we get. Now the structure differs in either of these 4. So it's not always in sulfur 30 or not always in Naxfoam. Maybe differ with the number of hydrogen bonds, in somewhere in the strength of the hydrogen bonds. So this is being differed. Now, as these 4 structures, either of these 4 items have been differed, as the potency changes, as the dilution changes, so it differs from the dilution to dilution or potency to potency. Now, this is a big challenge. It's not very easy, whatever the way I tell you. It's but we have there are a certain kind of instrumentations or the spectroscopical study that can only understand the difference between the hydrogen bonds or all these. So these differences can only be seen with the spectroscopy studies. Now, whatever Sukul Institute is engaged with the Raman spectroscopy and IR spectroscopy or infrared spectroscopy, and also fluorescence spectroscopy. Now, Raman spectroscopy has got mainly or basically the vibrational effect, the change in the vibrational of molecules. I'm talking about in a dilution or a potency, whatever. Now, this detects, the Raman spectroscopy only touches or only detects the vibration of molecules. So the vibration, in other words, the vibration of molecules changes as the potency changes or the dilution changes. So if you go for a dilution run with a Raman spectroscopy, you will find the changes in the vibration of the molecules. So thereby, you can understand, oh, this is sulfur 30 and this is Naxfoam 30. Only being the 2 molecules, carbon and hydrogen and oxygen. These are the 3. So they are the change. But the hydrogen bonds, oxygen and hydrogen, these are the bond hydrogen bond, that it differs with the vibrations. Now, if it comes with the fluorescence spectroscopy, though if you see separately oxygen or say separately now hydrogen are not a fluorescent material, so they are not fluorescent. But a Chinese group of scientists, they have recently discovered, is a remarkable discovery, what we believe, is that if hydrogen and alcohol and water joins together, then it gives you the fluorescence. Nor alcohol, neither the water is a fluorescent material. But if they join together, if they come together, they will give you the fluorescence. So thereby you can detect the fluorescence of these dilution or the fluorescence of the potency. The fluorescence can be detected or determined by the fluorescence spectroscopy, like what I said, that infrared fluorescence spectroscopy. So thereby it can be different with the sulfur or NAX or the other homeopathy medicines and the potencies. So these are the basic challenges as far as the basic research is concerned. Now, of course, there are a lot of things, clinical aspects of what I believe is not that much being challenged, or researchers who have been engaged with the clinical research are not facing a lot of these challenges. But of course, as a scientific field, because you have to answer in a scientific world, you have to publish your paper, good scientific paper. Non-homeopathic scientific paper is of course preferred because it's a science. So when you publish in a non-homeopathic journal and come into this with this data, a lot of questions will be asked to you. So you have to go through all the questions very correctly and with all the proofs that we have the data that have been found during the spectroscopical studies. So these are the challenges, of course, we are fitting. Sukul Institute has partially been done with this, differentiation between the potencies or the differentiation between the dilutions. And of course, there are a lot of things to do, and it's a big interest of all the scientists. They're working with this high dilution pharmacology. So that has been a great way to go further. And I believe that in next years or so, these things are the kind of hydrogen bonds will be exactly pinpointed to go further into the differentiation of potencies or the dilutions. So thank you very much for your patient hearing, and I'm thankful for inviting me, and really thankful to Dr. Sahani, who has been organizing such kind of seminars for so many years. I think even in COVID days, I spoke to this evening. So thanks once again.
Thank you so much, sir, for your valuable experience sharing and for highlighting the basic challenges we face in homeopathic research. Now, my next question is to Dr. KHURANA, sir. Sir, how can we ensure reproducibility in laboratory research on homeopathy when ultra dilutions are highly sensitive to preparation methods?
Yeah, it's quite a sensitive area in the sense that standardization of homeopathic preparation is a little variable, and they differ from pharmacopoeia to pharmacopoeia. US pharmacopoeia, European pharmacopoeia, or Indian pharmacopoeia. There are small differences in preparation of homeopathic medicines. Yes, they are sensitive in that way. So reproducibility is an issue. That will bring only laboratory or bench side studies. It's not difficult to reproduce those studies. You have to have a proper controls, and every time you can work out with different kind of preparations. From batch to batch, variations are there. Sometimes there are contaminations in the preparation stage, or there could be a microbial contamination, or even dust particles, they may enter into those medicines. So they do bring on some kind of different kind of effects. Yes, standardization of preparation of homeopathic medicine is a core area where we have to work a lot, in the sense that if you want to replicate the study and you desire that same kind of effect should be seen every time, then you have to standardize those processes. Experimental models are already standardized. There's no difference between them. You can antiviral effect or antibacterial effect you want to see on a laboratory model or animal model. The models are standardized. The only thing, the quality of preparation, the quality of medicine. Every time, the next time when you want to reproduce that same medicine in a different model or at a different study center, you have to see that that medicine which you have received is actually same. Now the question is, how you identify that the medicine there's no spurious content or there's original particles are there, and that's why CCRH worked on that area, and that's why we worked on the nanomedicine that time. That can be an answer whether we can have some kind of a standardization that these medicines contains a certain element which we want to see to be there in every potency. And the nanomedicine or nano technology has helped us in answering those questions.
Okay, thank you, sir. Now my next question is to Dr. K.C. Muralidharan, sir. Sir, how can a patient-reported outcomes and qualitative symptom analysis be integrated into evidence-based frameworks?
First of all, when we are collecting such qualitative data, so we have to think that the data should be properly recorded. So that quality of the data should be maintained properly. Then only we will be able to produce those data into a good outcome. Otherwise, we will not be able to do. Because generally this context belongs to some sort of qualitative research or clinical research mostly. And when we are looking at the research like psychiatry, suppose psychiatry, most of the time you are getting some sort of patient reported outcome and the qualitative symptoms. In such conditions, we have to use the standardized scale, assessment scale, for getting this data into a quantified format. The symbol is what we are using it. VAS score is one of the pain score we are using it. So we are converting those pain score into a quantifiable outcome through some sort of visual analog scale. So this type of methods only possible. You can convert those qualitative data into quantitative data. And some area where we can take patient-centered feedback, like in Likert scale. So we can use those Likert scale for patient-centered outcome, patient-centered feedback. You can collect it and you can convert into quantifiable data. And modern technologies like natural language processing also can be used for such type of techniques. But the core of the subject is core of this activity should be the data recording. So what I request all these practitioners, whatever you are doing, because the simple way of recording in practice, as per the practitioner's perspective is concerned. So generally they do case report publication. So that is a common things people do it. But whenever doing such common things, such a common thing also, you have to have proper recorded data. So then only we'll be able to convert those into a evidence-based system. Otherwise, a partial data or a manipulated data will lead to a wrong impression and a wrong outcome. So always keep a quality data is the main focus on this. So such scoring mechanism, assessment scales can be standardized. Assessment scales can be utilized for converting those qualitative data into quantifiable data analysis. Thank you.
Yes, sir. I fully agree that the quality of data as well as the appropriate use of standardized scales and tools is of paramount importance. Thank you, sir. Now, my next question is to Dr. Sukul, sir. Sir, how can existing public health data sets be mined to identify patterns where homeopathy has shown measurable benefit?
And the public data, both of the clinical and the basic research of the data, are being found in the published papers only. So there are the domestic or the national journals as well as the international journals. That doesn't always mean that these papers should be published in a homeopathic journal or a non-homeopathic journal. So a good work can be published in a homeopathic or non-homeopathic journals. Now, regarding the researcher, it's an access. So because the researcher, all the researchers are engaged in a particular, field. So the researcher are not finding so many aspects of research of homeopathy. So they are engaged in a particular field. So those particular field can be searched out, because with the you can search with the name of the scientist that you know already, or you know your work is being done with those scientists or those countries, and there are some other kind of search methods. So nowadays, of course, the AI tools is a very excellent helpful method of searching those data. Now these data can be easily accessed partially, of course, with the to the scientists, those who really need it, because first you will get a journal published that is there in the introduction part. You will not get the full paper until and unless you are asked for or you are being paid for it. Now, there are, of course, certain universities or institutions, they are accesses with so many journals of their related field, and so those can be easily accessed to those to the scientists that they are engaged into. So these, of course, are a public data, and this can be used to strengthen the field of the homeopathic research in the way that they are being referred. Now, whatever in course of basic research, we take the previous or similar or related researches from any part of the world, and take the papers and we cite them, we make them as a reference, and we name all the papers with their authors, with the topic of the papers, and then we publish it. So it will be another public data, so that will be being carried forward. So a little bit of change will be there of the new paper, and that will be from this previous paper that might be worked in some other countries, but the topic should be remain the same. So all the scientists, they are engaged in a particular topic that had been engaged for maybe several years now. There might be shift, but these can be a good source of public data, I think, other than, of course, the government. They keep their data in regard to the clinical research. Thank you.
Thank you so much, sir. My one more question to Dr. Khurana, sir. Sir, what role can nanotechnology and advanced spectroscopy play in validating the presence of biologically active components in homeopathic remedies?
I think regarding spectroscopy, Dr. Sukul has already explained some of his work. His father and he himself with some other institutes, they have been working on that particular spectroscopic spectrums of homeopathic medicine. But regarding
Yes, sir.
nanotechnology, we have already worked on IIT Mumbai and IIT Delhi at that point of time. And those articles are published. Now they have been good. These have been good studies where we actually demonstrated the presence of nanoparticles even in 30 to 100 potencies also. The only thing, how to explain that, how these particles are there, a theoretical explanation has been given by Dr. Bellare in his article also regarding the preparation of these development of these nanoparticles which travel from one potency to another because of the surface tension and bubbling effect which comes out because of this potentization or attenuations. And that is when the particles, they travel to the surface, they are taken up to the next part of the potency. When we take one part from the previous potency and 99 parts of the vehicle control. So in that way, we have demonstrated how these particles, they collide with each other, they reduce in size, and they become nano-sized. And these nano-sized particles are even, we could find even, we could quantify them also in 200 potency. And I think 1500 ml, he could demonstrate that there is some picogram of particular medicine. He worked on metals basically, zincomet, aromat, and argentum metallicum, zinc, Ifph metallicum, all those medicines he has worked upon. This article is in public domain, and any reader who is interested to see. And this nanotechnology is going to reshape homeopathy. These kind of studies are replicated in other labs where they can also demonstrate that these nanoparticles are there. And that's why the second question which you asked about the reproducibility, that is quite pertinent in the way that, yes, homeopathic preparations have to be standardized in terms of availability of those nanoparticles, how much quantities are there, that active component for which we are looking towards action of homeopathic medicines, whether they are there in our high dilutions, and that action can be demonstrated through those. When you can tag those nanoparticles with some radioactive substance, then look into the active transport of those materials to the target organ or the target cells at that point of time. But first of all, with this nanotechnology method, if these homeopathic medicines preparation is converting these original substance into a nano-sized particles, and they are available in 200. These kind of studies have to be replicated and reproduced, and that will make a big change, and that will bring a lot of scientific acceptance in homeopathy that these are not mere placebo, they have some biological active substance in high potencies.
Thank you so much, sir. My next question to Dr. Muralidharan, sir. Sir, what strategies can address the challenge of small sample sizes and long follow-up periods in homeopathic clinical research?
So my another question is why you are asking for a small sample size? That's the question. Because when we are conducting a research, so we will be deciding this sample size as such, always. And what I am talking about this, when we are discussing about this small sample size, generally this happens with rare disease conditions where we are not going to get any sample size as such. In such conditions, we don't do any RCT, randomized control trial, because we are not going to get any type of outcome on, because we are not going to get the required sample size for a particular condition. Even for exploratory also, we will not get such sample size. It is a rare disease, maybe 1 million, one patient or in a million population. So, which is not possible. So we have to depend on the experience of these case reports, whatever published available. That is the thing. And secondly, regarding this small sample size and long follow-up period in homeopathic clinical trial. And long follow-up period is always a issue for any type of research conditions, because we will be mixing up with a lot of other data, because the patient, even geographical changes also can influence his outcome. Even the patient data, his basic data also may change. His height, weight, all other things will be changing in a long-term effect, actually. So, but we can go for some Bayesian statistical method. So you have to think about the different statistical method for that. Rather than in a small sample size, we could conduct some research and getting something. And another issue with the small sample size is that actually there are a lot of underpowered studies will be coming out. So the studies will not be having that type of rich power to disseminate those data, because ultimately what is happening when there is a power of study is going down, definitely it is going to affect the outcome. Okay, so it is going to affect the outcome and it is giving a false positive result mostly, because the power of the study itself is not up to that mark. In such conditions, when you are going for some sort of meta-analysis and other things, definitely will not be getting a positive findings. You can see that there are studies related to, you can say, it is because of some sort of wrong selection of the medicine is also. That is one of the component we are not getting the actual outcome of research studies. These are some of the challenges actually out of this context I am telling, some of the challenges we are getting. But a small sample size can be treated by, I've already discussed in the first part, I think, N-of-1 trials or multi-center studies can be adopted to compensate those things. Then Bayesian models of statistical analysis can be undertaken. So that type of things can be undertaken and can reduce it. But I personally believe that that sample size, it is the feasibility of the study has to be first understood. Then only you take up the studies. So otherwise you don't take up the studies only for a few sample size for getting a very wide range of disease condition. when we are end up with a small sample size and get up with a inconclusive data will hamper homeopathy rather than research. Okay, thank you.
Thank you so much. Very aptly said. And now we are close to the conclude this session.